Hello, this is a video, the first video for the Biology 30 final exam review. So if you're getting ready for your final exam and you want to ask yourself some questions to see if you remember enough to be ready for the final exam for this course, then this is the video for you, I suppose. So I've arranged this as a bunch of questions to ask and talk about just so I don't forget. First, we have mutations can happen in many locations. So many different types of cells can, of course, suffer mutation. They can be exposed to mutagens, or those mutations can happen at random. Which location would result in descendants with mutations? So which types of cells would result in mutations that are heritable, that can be inherited from one generation to the next? So we know that if that happens in your finger or in your ear or in your liver, that's going to be mutations that last in your body but will not be passed on to the next generation. So this has to be mutations that are happening in your gonads because that's where you create gametes like sperm and eggs. The process of creating sperm and eggs is meiosis, and we could further narrow it down into creating sperm, spermatogenesis, or creating eggs, oogenesis, but these are both types of meiosis. For sexual reproduction, the advantages are, I think the biggest one is that you have bigger diversity, more variation in your offspring, which means you're better able to adapt to a changing environment. So this is because during sexual reproduction, during meiosis, we have an intentional kind of scrambling or mixing up of the chromosomes. Some alleles go to one offspring, some to another offspring. For sexual reproduction, we have some disadvantages too, like it costs more, it takes more ATP. It's possible that those advantages, those diversity, those adaptations, those differences are going to be harmful instead of beneficial. It takes time to locate a mate, and if something is working really well, then diversity is not very helpful. For asexual reproduction, you get something that is an advantage or a disadvantage, which is a population that is essentially clones, where the DNA is as close to identical as can be. This can be a good thing if you're already well fit to your environment, or a bad thing if your environment is changing and you can't adapt. For asexual reproduction, it is more efficient. It costs less ATP to do this process. You don't have to go find a mate. You don't have to build gametes. You can just skip right to budding or binary fission or however you're going to do that. For asexual reproduction, clonal population, that could be an advantage or a disadvantage. Another disadvantage could be you're less adapted or less adaptive, sorry, to changing environments. And also the speed of evolution, the speed of adaptation to your environment could be much faster in sexual reproduction, and generally is, than it is in asexual reproduction. For mitosis and meiosis, we've got a lot of steps. We know we have prophase, metaphase, anaphase, telophase for both of these. I'm just going to put this picture in here because that's better than my messy writing. In mitosis, in prophase, we start with our parent cell that has, it's 2N, it's diploid. We duplicate the chromosome. So one chromosome like that guy is now going to be split into two sister chromatids. This is still one chromosome because the naming is weird, but it's composed of twice as much stuff as before. So in prophase, we make our sister chromatids. In metaphase, they meet along that equator line, along the middle. During anaphase, they start to pull apart. Some go to this side and some go to that side. And during telophase, that's when we have the formation of the cell membrane around both of our daughter cells. We start with a 2N, a diploid cell. We end with a diploid cell. In mitosis, these two cells should be about equivalent to each other. In meiosis, We have prophase 1, 2, 3, and 4. In prophase 1, we have a similar thing. We have chromosome duplication. But during the chromosome duplication of meiosis during prophase 1, we make these tetrads, and the tetrads are groups of two pairs of sister chromatids that do crossing over. And crossing over, remember, was another way to increase genetic variation because we can have alleles from one part of the chromosome, so genes from one part of the chromosome, flipping to a different one. During metaphase 1, the same type of thing happens, only with twice as much material. So we're aligning at the equator. During anaphase and telophase, the same kind of thing happens. We are breaking apart, but once we have finished, we end up with still these sister chromatids remaining together that have to divide again. So that happens prophase, metaphase, anaphase, telophase 2. The difference between the two parts of meiosis and the one part of meiosis, the first stage and the second stage, is that in the second stage, we don't have the chromosome duplication happening. This means that in the end, our 2N cell is going to come down to a haploid cell, so diploid becoming haploid. So we skip the duplication process, which means that by the time we get our 4 daughter cells, we end up with 4 different ones. They're all different from each other, so they are not equivalent, and they're all haploid. If these were egg cells, 3 of them would be tiny polar bodies, and 1 of them would be a big healthy cell. If they were sperm, they would all be about the same. So in mitosis and meiosis, we have prophase, metaphase, anaphase, telophase, but in meiosis, we have it twice. And in the second part, we don't do chromosome duplication. When we have our nuclear membrane, so the membrane around our DNA, that is going to go away before, obviously, we can line up along the middle, because the middle of the cell isn't a place for the nucleus, or at least not that widely distributed. So the nuclear membrane disappears and reappears. It disappears during prophase as these things are getting ready to separate from each other, and reappears during anaphase or telophase. And kind of draw the line wherever we like there, but it reappears towards the end of both mitosis or meiosis. If we're looking at this and we want to know what the stages are, I think that's good. What kind of cells do we get by meiosis? We get haploid cells. We get, if most of our cells are diploid, so they start with a 2N cell, we go to an N cell. So if it's like 2 times 23 is 46, in the end we just get 23 for a sperm or an egg cell. And the 2 specific names for that process I already had up at the top, I guess, spermatogenesis and oogenesis. When we're looking at our DNA, this image shows up a lot. I'll do it in a color that actually appears. This image shows up a lot, something that looks like a big letter H or something with the centromere in the middle, one sister chromatid and another sister chromatid. This is a picture of one chromosome, but it's also a picture of 2 sister chromatids. If the picture was, I'm going to scratch out one of these, and the picture was just of this side, it would also be a picture of a chromosome, but only one chromatid. Didn't come up with the naming. I'm sorry. Down here we have a little animation playing that I stole from Amoeba Sisters, which shows fairly well that before interphase we've got 46 chromosomes and 46 chromatids. After interphase we double, we get ready for, we do the chromosome duplication, we double our chromatids. We still have 46 chromosomes, but we got 2 pairs of them now, like now in this picture. In meiosis, we are going to come down to half, so 23 chromosomes, but 46 chromatids. And after meiosis 2, we're going to divide this last one in half as well to come to 23 and 23. So the naming that this is a chromosome, but so is just half of it, is confusing, but I think you got it. The question, do all of your cells have all of your DNA present? Well, we know that our sperm cells or our egg cells have half they've got just one allele compared to the two that are in our somatic cells. So in your skin cells, in your brain cells, in your liver cells, in your kidney cells, these all would have a full copy of your DNA. Some cells don't, like sperm or egg cells. They've got a full copy of one allele, but not both alleles. And some cells don't have any DNA present, like your red blood cells. They're too busy carrying their oxygen around to do anything else. So over here, I have a couple more questions. What kind of structures are normally haploid? Sperm cells, egg cells, spores. If you're a plant, maybe. How do gametes end up with an abnormal number of chromosomes? This was a problem that occurred during this stage, so either pulling apart anaphase 1 or anaphase 2, where we have a problem with disjunction, breaking apart. So if we have a disjunction abnormality, if we're not pulling these things apart, this could lead to maybe having too many or too few copies of a chromosome. And if we were looking at uh, a cell's chromosomes and we wanted to see how those looked, if it was the correct number or not, then we would be looking at something like this, which is called a karyotype. So this one here is a karyotype. And karyotyping is a way of looking at chromosomes. Here we have an abnormality. The abnormality is that we have 3 copies of this chromosome here. This is, we can tell this person is female because they have 2 X chromosomes and no Y chromosome. But they have 3 copies of chromosome 21. So we could call this trisomy 21. I'm going to stop here. We're already 12 minutes in, and I think that that's as much of your attention as I could expect. I'll make another video for Unit 5 and another for Unit 6.